Researchers are currently testing 64 vaccines in clinical trials on humans. And 19 have reached the final stages of testing. At least 85 preclinical vaccines are under active investigation in animals. Here is a list of leading vaccines that have reached trials in humans, along with a selection of promising vaccines being tested in animals. Vaccines typically require years of research and testing before reaching the clinic, but in 2020, scientists embarked on a race to produce safe and effective coronavirus vaccines in record time. There are 4 categories of vaccines in clinical trials: nucleic acid, viral vector, protein subunit, and whole virus vaccine. Some of them try to smuggle the antigen into the body, others use the body's own cells to make the viral antigen. Nucleic acid vaccines use genetic material, either RNA or DNA, to provide cells with the instructions to make the antigen. In the case of COVID-19, this is usually the viral spike protein. The coronavirus is studded with proteins that it uses to enter human cells. These so-called spike proteins make a tempting target for potential vaccines and treatments. The vaccine uses messenger RNA, genetic material that our cells read to make proteins. Because of its transitory nature, there is zero risk of it integrating with our own genetic material. The molecule, called mRNA for short, is fragile and would be chopped to pieces by our natural enzymes if it were injected directly into the body. To protect their vaccine, Pfizer and Moderna wrap the mRNA in oily bubbles made of lipid nanoparticles. After injection, the vaccine particles bump into cells and fuse to them, releasing the mRNA. The cell's molecules read its sequence and build spike proteins. The mRNA from the vaccine is eventually destroyed by the cell, leaving no permanent trace. Some of the spike proteins form spikes that migrate to the surface of the cell and stick out their tips. The vaccinated cells also break up some of the proteins into fragments, which they present on their surface. These protruding spikes and spike protein fragments can then be recognized by the immune system. The viral vector vaccines are based on the virus's genetic instructions for building the spike protein. But unlike the Pfizer and Moderna vaccines, which store the instructions in single-stranded RNA, the viral vector vaccine uses double-stranded DNA. The modified adenovirus can enter cells but can't replicate inside them or cause illness. The Oxford-AstraZeneca researchers added the gene for the coronavirus spike protein to another virus called an adenovirus. Adenoviruses are common viruses that typically cause colds or flu-like symptoms. The Oxford team used a modified version of a chimpanzee adenovirus known as CHADOX1. The Johnson & Johnson researchers added the gene for the coronavirus spike protein to another virus called adenovirus 26. Sputnik V researchers added the gene for the coronavirus spike protein gene to 2 types of adenovirus, one called AD26 and one called AD5. Sputnik V comes out of decades of research on adenovirus-based vaccines. After vaccine is injected into a person's arm, the adenoviruses bump into cells and latch onto proteins on their surface. The cell engulfs the virus in a bubble and pulls it inside. Once inside, the adenovirus escapes from the bubble and travels to the nucleus, the chamber where the cell's DNA is stored. The adenovirus pushes its DNA into the nucleus. The adenovirus is engineered so it cannot make copies of itself, but the gene for the coronavirus spike protein can be read by the cell and copied into a mRNA molecule. The mRNA leaves the nucleus, and the cell's molecules read its sequence and begin assembling spike proteins. Building spike proteins process is similar to a nucleic acid vaccine, but adenovirus also provokes the immune system by switching on the cell's alarm systems. The cell sends out warning signals to activate immune cells nearby. By raising this alarm, adenovirus causes the immune system to react more strongly to the spike proteins. Let's compare nucleic acid versus viral vector vaccines. Immune response involves B cells and T cells. Well-established technology, minimal side effects risk. No live components, so no risk of the vaccine triggering disease. Strong immune response. Relatively easy to manufacture. Vaccine doesn't require ultra-cold storage. Booster shots may be required. Previous exposure to the vector could reduce effectiveness.